Olá, pessoal! Como estamos hoje? Uma sexta-feira, iniciando as nossas transmissões, finalizando a nossa primeira semana de estudos, mas nem por isso ainda vai ser levinho. Hoje de manhã nós teremos a presença do professor David Mann, e à tarde nós teremos a presença da professora Mariângela. A professora Mariângela vai falar sobre criptofícias e euglenoides, e o professor David Mann vai trabalhar, em geral, sobre o tema das diatomáceas, né? Diferentes técnicas aí aplicadas para o conhecimento das diatomáceas. Antes de mais nada, antes de começar a apresentação, eu gostaria de fazer um triste anúncio que, por questões técnicas, infelizmente, nós não teremos a nossa programação cultural. Isso, infelizmente, por conta de dificuldades técnicas, o quinteto não vai poder fazer a apresentação, tá bom? Mas, de repente, durante a nossa próxima semana, a gente consiga, então vamos ficar no aguardo. Então, eu estou anunciando já que eu estou tirando da programação, né, que estava programada aquela programação cultural para as nossas 19 horas, ok? Bom, falando agora do professor David. Eu sou um grande admirador do professor David, tá? Porque ele há bastante tempo desenvolve trabalhos realizados relacionados às diatomáceas, diferentes vertentes de estudos para a diatomácea, desde artigos taxonômicos até de biologia, filogenia. Ele é muito versátil nesse sentido, né? Ele desenvolve diferentes tipos de artigos mesmo, de ciclo de vida. É impressionante, assim. Então, ele tem sua formação na Universidade de Bristol, universidade lá da Inglaterra, apesar dele ser escocês. E aí ele fez o doutorado dele, inclusive o doutorado dele, mestrado, doutorado, também na Universidade de Bristol, certo? Inclusive, existe uma diferença, que ele fez o PhD na Universidade de Bristol em 1978, tá? E em 2006, ele fez o doutorado em ciência também na Universidade de Bristol, tá? Ele é detentor de uma série de prêmios e honrarias no currículo dele, inclusive por trabalhos bastante conhecidos. Ele é um dos coautores, por exemplo, do livro do Van den Heck, que a gente utiliza bastante, né? E também um dos coautores do livro da Bíblia de diatomáceas, que a gente utiliza bastante, né? Que a gente poderia dizer aí, que é o The Diatoms Biology and Morphology of the Genera. Aquele livro de 1990 e o Van den Heck de 1995. Ele já foi também o presidente da International Society for Diatom Research de 2010, entre 2010 e 2012, e atualmente ele tem trabalhado no Royal Botanical Garden Edinburgh como pesquisador associado. Ele já é aposentado, né, mas continua como pesquisador associado no Royal Botanical Garden. Então eu gostaria de convidar, né, isso mesmo, Lesilda, foi discípulo do Dr. Round, que é um dos autores do livro das diatomáceas, né, the Diatoms. And then I would like to invite our transmission Professor Dave Mann. Professor, it's an honor, it's a pleasure for me to receive you here with us in Brazil to see your presentation because, well, I was saying for our colleagues here that you work with a lot of tools for diatom studies, like taxonomy, phylogeny, life cycle, and ontogeny, morphology, and well, you have a lot of perspectives about diatoms. It's very interesting your studies, and for me, it's an honor to receive you here with us in this second edition of our event. Thank you.
Well, Thank you very much for the introduction, and really nice to be here. Unfortunately, I haven't been to any of the other talks this week. I'm actually, as it were, quarantined at the moment. I flew into Edinburgh last weekend, and then within 3 days, they tell me that I have to stay at home because I've been in contact with somebody with COVID. So I'm sitting at home rather than in the Royal Botanic Garden, Edinburgh, but we'll see how we go. I can't say hello in Portuguese, but in Catalan, bon dia tothom. It's very nice to be here. So I will now share my screen and we'll see how we go.
Okay, we can try to share the presentation now.
So hopefully you can see.
Yes, perfectly.
Perfect. Good.
You have the words.
Okay. So this is my title for today: Understanding Diatom Species, Shape, Sex, Smell and Sequences, and something about Metabar Coding. I've been thinking about diatoms for 45 years now, if my arithmetic is correct. And I hope I can convey something of what I think I've learned in that time, plus also all the things that we still don't know anything about. So first of all, in case this is not an audience that knows everything about diatoms already, what is a diatom? Well, this is probably what most people would think of as diatoms, a glass box basically in which a little cell lives, and it's a glass box which consists of two major pieces, one at one end of the cell and one at the other end of the cell. Here's one, here's the other, and a region in the middle which we call the girdle. So these two pieces, one at each end, are valves, and then you've got the girdle in the middle. In fact, actually, what most people think of as diatoms is not that kind of complete box. For most diatomists even, the box is just reduced to one of those valves. All people care about most of the time is those end pieces, the valves. So here we have a couple of those end pieces in the light microscope, and here we have a scanning electron micrograph of one of these end pieces. It's very important to realize these are not diatoms. They are pieces of diatoms. It's like saying, for example, that a bone is the same as a triceratops or a human being, or a feather is the same as the bird, or a mollusk shell is the mollusks. In fact, the diatom is the whole cell, of course, and the whole life cycle that it has from one end to another. So this is a closer approximation to what diatoms are. These are the living cells, and some of them have got lots of little chloroplasts inside, like these two here, and others have got much larger, smaller numbers of chloroplasts inside. And those chloroplasts are usually yellow-brown because of the presence of fucoxanthin in them, as well as the chlorophylls. What are diatoms in terms of their place in the tree of life? Well, they belong to a group called the Straminophiles, which also includes the brown algae and various other groups of yellow-brown algae. And they are more closely related to things that don't have chloroplasts, like some of the alveolates and some of the amoebae, than they are to green plants and red algae. I'm sure you're probably familiar with that. This tree of life is one of the more recent ones, and if you want to look it up afterwards, if you don't know this paper, it's on the references there. We know now what the diatom's closest relatives are, and they don't really look all that much like diatoms because they have a life cycle that involves a biflagellate cell, which was seen and then classified as a bolidophyte, a member of the Bolidophyceae. And then they have a stage in the life cycle that is silicified with these interesting looking scales around the outside, which were classified originally as parmales. These two stages belong together, and of course the only bit that is kind of really linked very easily to diatoms are these silica elements, which look a little bit like diatom valves and girdle bands, but they are arranged in a completely different way. What else is particular to diatoms? Well, first of all, we can think about the diatom cell division and life cycle, perhaps. They have a rather special method of cell division, and they have a rather special life cycle. So when you see a diatom dividing, one thing that is striking is that the old cell wall around the diatom does not disappear. It's not like some of the green algae, for example, that produce new daughter cells and throw away the old cell wall. No, the diatom keeps its cell wall, and it's inside that cell wall that new valves and new girdle bands are produced. So here's a dividing cell, just about to divide, partway through division and after cell division, and this cell here will be forming its new valves inside the old cell wall. Here's a diagram of that event happening. So the new valves are made within the parent cell wall. And that means, of course, that because the outside cell wall is really rather an inflexible silica structure, it is flexible to some extent, but often it causes the new valves that are formed to be smaller than the old ones. So you can see in this case here, this is the older part of the cell wall, and it is just that little bit bigger than this, which is the newer one formed after the last cell division. If this one fits inside that one and is therefore has to be usually that little bit smaller. Now, all this is familiar to any diatomist, but I think it's still a surprising thing to people who don't work much with diatoms. But the end effect is that during the life cycle, as the vegetative cells divide, gradually the cell size gets lesser and lesser. The valves become smaller and smaller, and they often change shape. Like in this one here, this cell with its kind of almost rounded profile is derived in the end from this rather longer rhombic cell at the beginning. Now that means, of course, that at some stage something's got to happen, because if it doesn't, then the diatom population is going to die out, because the cells will just keep on getting smaller. So as many of you will know, diatoms then go through a stage of size restitution. They get bigger, they expand again. So here we have got some small cells, and this particular cell here has given rise to an expanded large cell, which will then produce new valves and start the life cycle all over again. In some cases, the expansion phase is really quite interesting and complicated. So in this diatom, which is called Nichia inconspicua, the old cell here splits apart. It forms a little spherical cell within it, and then that spherical cell doesn't stay spherical in a nichia, but expands in two directions. Here it is just beginning to expand here, and here it's expanded a lot more until it gets to a maximum for that particular species, in which after which it produces new valves. Here's the old valves. This is going to produce new valves inside, and then the whole process of getting smaller will start again. So we can summarize the life cycle of your diatom, of your typical diatom. I say typical because this doesn't happen in every case. We still don't know the extent to which some diatoms avoid this process, but some do. But in the typical case, a diatom gets smaller, then it goes through meiosis and fertilization to produce a special cell, the spherical one that we were seeing in these last two cases. Not always spherical, but in the two examples I showed that it was. And then that cell expands to a maximum and then starts the process again. All of these cells, the vegetative cells, are diploid. Only the gametes are haploid. And this expansion stage is called an auxospore. Not every diatom cell can produce an auxospore. It's only the smallest ones that can produce an auxospore. So if we looked at a diatom population that is in the process of this auxosporulation, this getting bigger, we find that the middle-sized cells, the blue ones here, and the middle to small cells, they don't pair, they don't go through sexual reproduction, they don't auxosporulate. There is a critical size threshold above which cells cannot go through auxosporulation. Once they get to that critical threshold, in this case, at this point here, then some of them, and gradually a higher and higher proportion of them, can auxosporulate. They can go through the expansion phase. And after expansion, then they produce large cells. So this is the kind of spectrum of sizes that you get in a population as it's going through the size restitution stage. Let's just think briefly about diatom significance and occurrence. We'll get to diatom species not very long from now, but I thought we would just go through some reminders about what diatoms do and why they maybe are worth focusing on a bit. First thing is, as I calculated a long time ago, other people have kind of confirmed it, or perhaps they've pinched the idea. Diatoms, planktonic diatoms, perform about 20% of global photosynthesis, which is more than all the rainforests of the world. They do it because they are particularly abundant in the nutrient-rich areas of the oceans. So the polar regions, either the Arctic or in the convergence of the Antarctic, also in these upwelling regions of Africa, South America, and so on. They do things for other organisms. They're at the base of the food web, clearly, but they also perform other ecosystem services. For example, many of them growing abundantly on sediments stabilize those sediments via the polymeric substances that they produce for moving around. So quite a lot of diatoms are able to glide through, climb through sediments, and as they do that, they leave behind polymeric materials which glue the surface of the sediments together. Others do strange things like growing in the surf zones of the oceans. So in South Africa, South America, and this one in Edinburgh, you can find these surf zone communities. Very few species, but quite dramatic in what they produce. The foams and scums of diatoms that look particularly dirty, but are just because they've got brown colorations in the cells. Others grow attached. So there are some that grow attached to sand grains, these episamic diatoms. Others grow attached to algae or other plants, epiphytic diatoms. And some of them are rather surprising. This also shows a diatom. And those of you who are thinking, but that's a dinoflagellate, well, yes, you're quite right. But this is a dinoflagellate that is only able to live basically because it has a diatom inside it. So all these golden brown structures inside the cell here are endosymbiotic diatoms, or at least they're derived from endosymbiotic diatoms. These cannot anymore live outside the dinoflagellate. And the dinoflagellate's own chloroplast is reduced to a red eye spot. So this is a Durinschia, and we are particularly interested in those at the moment because they illustrate stages in how chloroplasts can evolve. Diatoms also do some nasty things. They produce, some of them produce domoic acid. Most of the species that we know that do this are members of the genus Pseudonychia. They produce domoic acid, which causes amnesic shellfish poisoning and killed a few people in Canada in the 1980s. And since then has been a main focus for people monitoring the health of coastal ecosystems and the safety of mollusks for eating. Diatoms, on the other hand, are extremely useful for biomonitoring because they have that silica shell around the outside. A whole industry of biomonitoring has developed, with people taking samples from rocks or other substrates in rivers and lakes and seeing which species are present and converting that knowledge into an assessment of ecological status. So I don't know what the position is in Brazil in this regard. I would be very interested to know more, but certainly in Europe and North America, diatoms have been used extensively for some time for establishing whether the ecological status of rivers and lakes is good or not so good or just plain poor. And nowadays that's moving from a microscope-based system to a system based on metabar coding, DNA metabar coding. And we'll come back to metabar coding later, but not for biomonitoring. Okay, so for those of you who are perhaps less familiar with diatoms, I hope that has given you just a few indicators about the kinds of things that characterize a diatom and some of the things that are unique about them. So now I want to turn to diatom diversity, and first of all to talk a little bit about the main relationships within the diatom group, and then to go on to the main focus, which will be species. So here's a picture of all the different kinds of valves that diatoms produce, different species, different genera. And when you look at that diversity and you start to try to kind of understand it and categorize it, you see that there are basically two main structural types, if you like, in the diatoms that have been recognized for over a century, called centrics and pennates. Some of you may now be getting worried that I haven't got up to date with what's been going on, but I'll come back to that in a moment. In terms of the overall appearance and structure of the diatom cell, there are these two groups, and let's just see what characterizes them. Centrics, and here's an example of a centric on the left here, have a pattern that is basically radial. We have ribs radiating out from the center of the valve, and at the center there is a little ring, what we call an annulus, just Latin for ring. And that subtends the whole system of radiating ribs of the diatom. So that's what the centric organization is like. And then there is a pennate system of organization, which consists of an axis, a rib that runs from one end of the cell to the other, which is subtending ribs on each side. So you have a bilateral pattern arrangement organized about this axis, the sternum it's called, in the middle. So you have those two basic types of organization of the valve. And that agrees with some other differences. For example, there is the life cycle. I showed you a summary of the life cycle earlier, and this is, in fact, was a diagram based on centric diatoms, which have a stage in which the sexual reproduction takes place, and sexual reproduction takes place through oogamy. We get the formation of little flagellate cells, a sperm, which fertilize a much larger egg cell. So here's an egg cell, and here's a sperm. The sperm have one anterior flagellum with little hairs on it, like all the other members of the Straminapile group. They are characterized by having one flagellum with hairs, and if they have a second flagellum, that second flagellum does not have hairs. Okay, so they have oogamous sexual reproduction in this type of diatom, in this centric kind of diatom. What about the pennate diatoms? Well, no, they don't form any flagella at all at any stage in their life cycle. In this case, cells basically form large gametes, which are more or less the same in each of the cells that pairs. This particular diatom, Salaphora oldriki, which I will come back to later for other reasons. This is a vegetative cell. The cells come together as a result of a pairing process, which again we'll come back to later. They go through meiosis, and each cell produces, in this case, one large gamete, and the large gamete from one cell moves into the large gamete of the other cell and fuses with it, and then they produce an auxospore. So here we have no flagella. We have large gametes that are basically the same size. They are kind of isogametes, even if they behave differently. This one, for example, one of the gametes is active and one of them is passive. There's lots of variations on this theme in pennates, but the basic process is the same. They fuse, and then we get our expansion as in the centric group. Okay, so there are differences in structure in the life cycle. There are also differences in the mating system. Basically, centric diatoms are homothallic. That means the life cycle can be completed by a single clone. There are no separate mating types. Whereas pennates, not always, but many, and probably primitively, they are heterothallic. The life cycle can therefore be completed in these only by a pair of compatible clones. So in this case here, we know subsequently to some experimental work that this cell has a different mating type to this one. And it's only by a pairing between different mating types that you can go through to form an auxospore. Okay, so we've got two main kinds of diatoms differing in pattern organization, mode of sexual reproduction, the ability to form flagellate cells, the mating system, and also there is a difference to some extent in chloroplast number and arrangement. The centrics tend to have lots of small chloroplasts, whereas the pennates tend to have one or two or a very small number of chloroplasts. Not surprisingly then, when you look at the classifications that people have evolved over the years to deal with diatom variation, to recognize what they think are the major groups, in the earlier ones, it was more or less uniformly a separation between the centrics and the pennates. They were put in two major groups. So the first really comprehensive classification of all diatoms was by Georg Carsten in 1928 in Die natürlichen Pflanzenfamilien, and he had two large groups, and this classification was used basically by most people and through to about 1990. So this is his classification. You have these two major groups, the centrales and the penales, and then a series of smaller groups to recognize the variation in each one. In 1990, we changed that a little bit. I was part of a book that some of you may know, focusing on the diatoms. And in that book, and I now actually don't really remember very well the rationale. Looking back, it seems a bit stupid, but perhaps that's with the benefit of hindsight. We recognized three groups because we split the pennates into two. We recognized one group for these pennate diatoms that don't have any special structure along the center of the cell, and we recognize separate a group of the pennates that have a raphe system and are therefore able to move, because the raphe system is a remarkable invention of a large group of diatoms that enables them to move around without flagella or any pseudopodia or anything like that. So we recognize centrics, araphid pennates, and raphid pennates. And this is our classification. Here are the three groups: the centrics, the araphid pennates, the ones that don't have a raphe, and the raphid pennates. I'm sorry that I call it raphid and araphid rather than raphid and araphid. I realize that my pronunciation is distinctly not as it should be, but the Anglo-Saxons, I'm afraid, mispronounce everything to do with Latin. So we divided diatoms into 3 groups. Karsten divided diatoms into 2 main groups based on the shape, structure, and pattern of the valve, the pattern of sexual reproduction, and various other things, which all seems to make sense from a functional ecological perspective. The trouble is that it's wrong. And we found out that it's wrong basically through the molecular phylogenetics that have been done during the last 25 to 30 years. The first person to get to grips with the diatoms in a molecular phylogenetic context was Linda Medlin, who basically invented the system of using 18S ribosomal DNA for looking at relationships in diatoms and other eukaryotes. Later, Ed Theriot and his team added a lot to our knowledge, working with a 3 gene system, 18S ribosomal DNA, RBCL, and PSBC, and people are now moving on to use the whole plastid genome or even more of the genome to work out relationships. This is the molecular phylogenetic revolution, and it has completely transformed how we look at diatom relationships. So when we get a tree from, for example, a 3-gene set, this is a tree that is kind of simplified from a paper in 2015 by Ed Theriot's group, we find relationships are like this rather than the 2 or 3 group system that I've just been describing. So instead of 1 group in the centrics, we find perhaps 6 major groups. The araphid pennates are not 1 group, but 2, and perhaps more, but at the moment it seems it's 2. And the raphid pennates are the only group of the original major groups that survives as a separate entity that is monophyletic and justifies being given taxonomic status, whatever your philosophy. A couple of years ago, I tried to codify this into names. There's no reason why you should believe that it's true or accept that it's the best system. It's just a suggestion based on the molecular data so far. And in that, I recognize a series of subdivisions within diatoms. Here are the centric ones here, and then the pennates, and some of the centrics are in a separate group. If we look at the Bacillariophytina, in detail, we find certain groups of centric diatoms and the pennate diatoms here. And then if we look at the pennate diatoms, inside them we have our araphid pennates and our raphid pennates. So this is, I think, the only attempt that has been made to make a formal classification out of the advances that molecular phylogenetics have provided. It's almost certainly wrong, but at the moment it's the best that I can do. Why does it matter? Well, it matters partly because people need a system of higher taxa, of classes, of orders, of subclasses, of divisions, subdivisions, and so on, to database data in a sensible way. And if you go, for example, to GenBank at the moment, you will find that the classification that they're using is completely out of date. It reflects what was thought as a result of the first molecular investigations, and it does not correspond to what is now known. So we've got all kinds of strange groups here, and this is not a coherent phylogenetic classification. And as a result of that, if you use the GenBank classification to, for example, make diversity estimates of particular types of diatom, you will get a very strange picture. And if you use their classification to interpret metabar coding data, it also will come up with some very strange results. Okay, now I want to move on to species. My plan, by the way, is to stop in about 15 minutes time and let you get your breath if you are finding I'm going a bit fast. Then we'll have a break, and then we'll come back and continue. And at the end, of course, I want to give you opportunity to ask questions or comment and so on. But we'll just press on now, if you don't mind, to think about species. I like this slide. It's nothing to do with diatoms, but it kind of gives a perspective on, as you will see, on diatoms. There is something rather strange about diatoms and their diversity, which in a way is a bit like beetles. So there was a prominent geneticist of the middle of the 20th century called J.B.S. Haldane, and he is supposed to have said, when he was asked about what God liked, what God's intentions were when creating organisms, he said, on being asked what one could conclude about the nature of the creator, about God, from a study of his creation, Haldane is supposed to have said an inordinate fondness for beetles, because as we all know, there are millions of beetle species on the earth. Well, what about diatoms then? Well, there are a lot of diatom species even now. And what I want to explore is whether that's an artifact, the truth, or an exaggeration. Diatom species, what are they? They seem to be a lot of them. Does it matter whether we have a good idea of species? Well, from a practical point of view, for biomonitoring, for example, or for the health of people, identifying species correctly is important. If, for example, we don't identify pseudonychia species correctly, we may have give problems to the aquaculture industry that are completely unnecessary. If we tell them that there are lots of poisonous pseudonychia species present, they may close their fishery, whereas in fact the pseudonychia species that are present may be entirely innocuous, harmless. Biomonitoring, if we get our species wrong, then we may tell water managers, river managers, that their river is filthy and dirty and has to be cleaned up, when in fact it's perfectly fine. The problem with diatom taxonomy is that it changes with time, and it changes in quite alarming ways. So at one time you will find that people are saying everything's the same. There are very few diatom species. There's a lot of variation in each one. And at other times people will say, no, there are lots and lots of diatom species. Everything you see, almost everything you see is different. And this kind of swing of the pendulum has been going on ever since diatoms started to be classified at the beginning of the 19th century. So, for example, in 1856, we had a fairly clear idea, apparently, of what diatom species are. This was before Darwin, and basically what diatom taxonomists were doing were looking to see which of the variations that they saw were genuine species that God had created at the beginning and had particular characteristics that were unchangeable because those characteristics had been given them right from the beginning. So you find a very clean taxonomy at that time. There were no infraspecific taxa. Variation in essential characters, in important characters, if you like, it allowed people to infer species boundaries. These species were products of the original creative act, whereas variation in inessential characters did not really need to be recognized in any way. So, in a way, there was a kind of lumping, and that was quite extreme in some cases. For example, this worker, Gregory, again, just before The Origin of Species was published, considered that all of these different things that you see here were likely the same species. Because as he looked at the variation, it just seemed so continuous and so bewildering and so complete that he could not, with any confidence, say that there were species boundaries there. So he was inclined to put all of these together in this taxon navicular varians, a rather good name. Then along came Darwin, and in a way he took the heat out of any argument about what species were, because despite the fact that he called his book The Origin of Species, he didn't really have a concept of species at all, because he thought that species were arbitrary inventions of mankind, that basically they had no special status. They were not essentially different from what other people would call varieties. They're just basically an arbitrary level in a complete spectrum of different variants. So in a way, people were set free to do what they liked, and most of them liked the idea of splitting. I think everybody likes to give things a name themselves. So people were set free to call things species if they wanted to, or call them varieties if they wanted to. And this was a time of expeditions, of explorations. People were going to new areas for the first time, collecting diatoms for the first time, and they had the freedom, if you like, to call any new variation that they saw either a new variety or a new species. And they didn't worry too much about which of those they used. The end result is that we have a huge number of taxa at species level, variety level, form level, which are now a huge and possibly impossible problem. We need somehow to try to sort these things out, to typify them, to work out exactly what people were looking at when they described these species, varieties, and forms. And in many cases, we don't have good material that's left. And there are such huge numbers of these taxa that it is questionable whether anybody will be able to work through them and work out exactly what they are. So I'm sure we are now describing a series of new species that have actually been described before, and nobody is going to have the time and energy to sort them out. Okay, is this proliferation, this description of large numbers of species and varieties and forms justified? Well, when I started in diatoms, and I'm afraid I have degenerated quite a lot since the early days when I started diatom studies, so this is me about 40 years ago. When I started, I was inclined to the lumping idea, that the idea that things had been over-described. There were too many diatom taxa. So when I was looking in my PhD and just after at some diatoms like this one, Hantia marina, I was struck that there was quite a lot of variation within what was then being called a single species. And I was inclined to think that this was real. In other words, that species are very variable things and that you couldn't distinguish meaningful varieties or any other taxa within them, let alone split them up into separate species. So here's our Hantia marina, and you from one particular beach, and you can see there's a small one here and a bigger one here. Well, you might think that was part of the life cycle, but it can't be actually, because this one has quite wide striations. They're quite far apart. Notice the spacing of all these little structures at the edge here, and this one has much more closely spaced structures. And we know from laboratory experiments that this kind of thing does not happen during the life cycle of a single clone. So we've got diversity there that is genotypic. And we can see this at various localities. So this is from one location in Scotland here, this yellow, orangey color. And this is a population of what is supposed to be the same species from a different location And at each location, there's a larger, more coarsely structured one and a more delicate one. And that correlates with various other characteristics. So the coarse one has slightly different holes through the valve. These are the holes through the valve. You can see that you've got a nice reniform structure here and simple pores in the other one that's living alongside it in the same place. So this is the coarse one. This is the finer one found at the same site. These are the two at the other site that I was mentioning, and they've got different pores to the ones in Cornwall. And they vary also in the substructure of these pores. This one has got these lovely organized rows of tiny 5 nanometer holes through the membrane here, the silica membrane, and this one's got a more irregular arrangement. They also vary in how the reef structure is organized. So at these two localities, you have two variants that differ from each other, and they differ from the ones in the other locality as well. So we have from two places 4 different variants. And if you go to a different locality, like Edinburgh, you find a different 2 or more variants. So what happens is if you gradually look at more and more and more samples, what seems at first to be a very clear-cut picture that you have 2 different types at one locality and 2 different types at a different locality. By the time you put lots and lots of localities together, you start to see what seems to be a continuous pattern of variation in this species. And that's just 3 samples in this graph here, but as you fill in more and more, you find that the picture just gets more and more complicated. This is a species group that I have spent a lot of time on, and these are valves from a series of different ponds and lakes and rivers in the UK and elsewhere, Belgium, Africa, Australia. And you can see that although if you were to look at one place, you might perhaps find a nice separation between something like this and something like this, by the time you sample lots and lots of different places, you begin to lose sight of a nice convincing boundary between separate taxa. So are these really the same as these? Well, we don't know, because they may be connected by a series of intermediate forms between this one and this one and this one and so on. So in 1963, in relation to this particular species group or single species, Robert Ross said, when a large number of populations are examined, the various forms integrate so much, there's so much connection between them morphologically, that there seems little justification for recognizing them taxonomically at any level. So that looked like an invitation to lumping. If you look at enough populations from enough different places, it looks as though there is no boundary between the variation that you find, and so perhaps there are very few species and each of them is very variable. That was reflected in the classifications that were produced. So one of the major works that is used for the identification of diatoms in Europe and to a lesser extent elsewhere in the world is the series of 5 volumes by Kurt Kramer and Horst Lange-Berthelot. They were published between 1986 and the middle 1990s. They covered every group of freshwater diatoms in Europe, and their taxonomy is basically a coarse-grained one. They lumped things together. So all of these, for example, were recognized as a single species, maybe with some varieties, but basically one species. And that same philosophy is seen in their treatments of other groups, for example, the group Nitzschia. And I think we'll stop there and have a break, and then I will pick up the pieces and take you through how things are changing, and then examine to what extent we can get some independent evidence about whether species are large or basically quite small. So I'm going to come out of my presentation now and hand over to
15 minutes?
Do you think about 10 or 15 minutes break? Would that be about right? I think.
Ok, it's ok. Então, pessoal, nós voltamos dentro de 15 minutinhos, tá bom? Fiquem à vontade aí, tomem um cafezinho, descansem um pouco, logo nós voltamos. Até mais. Olá, pessoal, estamos de volta. Welcome again, David. Let's start your presentation.
Okay. Okay, you see?
Yeah.
Okay, good.
Yeah, perfect.
Well, I can't see my audience, which is kind of strange, but I suppose we get used to that these days with presentations. Okay, so
David,
yeah,
David, sorry. Can you occult the bear located behind bellow? Yes, yes, good.
this, yeah,
Yes,
okay, will do.
perfect.
Okay, so we got as far as thinking that up till the 1980s, there was a kind of trend to putting species together, and this is a slide I think that expresses that pretty well. In the reclassification of some nichier species, these are all nichier species that you can see here, by Langebetlo and Simonsen in 1978. That was just as I was finishing my PhD. All of these different things got put together. So all these species that are listed at the bottom here were made synonyms of one, Nichiagandus hymiensis. But within a couple of decades, the things were moving back in the other direction. So for example, just to show what happened over 12 years with a particular group of 3 species, at first, these two were considered to be the same, but different from Nichia inconspicua. And 2 years later, all of them were made the same. 10 years later, 2 of them were separated again. So we're back to 1976. And then later in 1988, they were all 3 recognized as separate. Nichia recti robusta was described by Huschtit in 1950 as a separate species. 1978, no status for it. It was put into Nichia recta. 1988, it was pulled out as a zipper. I mean, in other words, a kind of race. Something that you don't recognize taxonomically, but you realize has some kind of existence. And then in 1996, it reemerges as a species. Nowadays, there are huge numbers of new species being described in phytotaxa and in separate books and elsewhere. So we've got this problem. What is the truth? Should we be splitting? Should we be lumping? It's a very difficult problem, and the solution is probably different in different groups and species of diatoms. What can we use as kind of criteria for trying to sort out this mess? Are there 1,000 or 10,000 or 100,000 species? Is it all a matter of fashion? Well, Darwin did not do us a service in many respects. As I said before, he didn't really believe that species have any particular objective existence. They are just part of a spectrum. In some ways, of course, this is true because evolution is a process. It doesn't happen well, it does happen sometimes in jerks, in kind of quantum bursts of evolution, but it is a process, and you don't expect a species to arise completely separate in one go. It's going to be a process by which it arises, a process of speciation. But I think it's true for most people that species have a different kind of status, and it's not just an arbitrary decision. There is a truth in species, I believe. Not everybody would agree. And this is because if you look at nature, you look at living organisms, you find that variation is discontinuous and non-random. So you find that different characteristics are kind of inherited together, and variation is, if you like, hierarchical. You can put things in a nested series, but there is a limit to that nested series. You go down from divisions to classes to orders to families, and everything is, if you like, neat. And then you get to a level where things fall apart. There's a boundary between hierarchical and non-hierarchical variation. variation is essentially hierarchical among species and non-hierarchical within them. And no other taxonomic category corresponds to a boundary of this kind. And basically, there is a mechanism which some people don't believe in, but I believe in, and I think there is good evidence for it, and population geneticists basically assume it. A mechanism that integrates populations and leads to this, the formation of this boundary between hierarchical and non-hierarchical variation. And this mechanism is basically gene flow, brought about by sexual reproduction, in conjunction with dispersal and migration. Now, this kind of emphasis on gene flow and so on, of course, comes from the new synthesis, as it's called, from the 1930s onwards. It's associated to a large extent with the names of Dobzhansky and Mayer. And they focused on genetics, populations, gene flow, and the idea of species as reproductively isolated units of biodiversity. So genes can flow between individuals of a species, but they can't flow, or they can't flow freely between members of other species. Putting it slightly more modernly, as it were, and taking some of the problems of the biological species concept, this idea of reproductivization, out of it, we come to a concept, I think it is very well explained and so on, by de Kerov in 2007. Speciation is the process by which one separately evolving set of populations splits into two separately evolving sets of populations. And that's a process, and the characteristics that we associate often with species develop at various times and in various orders in different groups. So the idea of species is it's the result of a process of splitting, which may not be complete in some cases. And you get the progressive acquisition of particular characteristics. Species gradually or quickly become phonetically distinct. We can notice them as different in some way. They accumulate fixed autapomorphies, in other words, characteristics that are unique to them. They achieve genetic coalescence, which I'm not going to try to explain. They achieve in time intrinsic reproductive isolation. So no matter what you put, how you put things together, they will never interbreed with each other. In time, they get niche separation. So they're ecologically different, perhaps. So all these are kinds of species characteristics, but they are not all necessary for this distinction of species. These characteristics develop successively and in different orders in different groups. And the order in which each species property is acquired is lineage specific. But we can use any or all of these characters to give us insights into what are genuinely different species and which are not. So, okay, I showed you this picture before. How many species are there here? How can we find out? Is it true that when a large number of populations are examined, the various forms so integrate that we can't distinguish any species among these different forms? Okay, well, let us apply some of the ideas from the biological species concept and de Kerov's modification of it and see what we come up with. So let's take Selaphora oldriki, for example, and explore it a little bit. This is the process by which sexual reproduction takes place. What if we mix different populations together and see what they do? I came across this entirely by accident. I was not aiming to study species concepts in diatoms, but in the early 1980s, when we began work on our book about diatom genera and trying to come up with a kind of comprehensive account of the diatoms, which we published in 1990. In the process of that, I looked at a lot of diatoms, basically just to see what kind of chloroplasts they had and to understand them better from the point of view of the taxonomy of diatom genera. But in the process of that, I just by accident started seeing sexually reproducing diatoms. And this is one of the first pictures that I took when I was working on this. And it's a picture that shows two of these morphological variants of Salaphora. Then it was one species, Salaphora pupula. There's one here that's a bit smaller and with a kind of lanceolate shape, and there's one here that's a bit bigger and it has a more linear shape. And you'll notice that these three are mating with each other. They're very close, but they are separated from this other group of four here, which are this morphological variant that's a little bit different. And there's a third variant here, which is a bit bigger than this one, and that's not interested in either of those two. So these are reproducing sexually, so are those. This one isn't. So there's something going on that separates them. And I followed that in detail, and we did laboratory work later to test our field-based observations, and found that basically each of these variants that were growing sympatrically in the same pond were just not interested in each other. You could put them into the same pot and they wouldn't interbreed. You could watch them in semi-natural populations brought in from the pond and they weren't interested. So this is a case here. We've got length versus width of the cells, and plotted on there are the ones that are mating. So a line indicates that one cell here is mating with another cell, and you can see that there are two different mating populations that are remaining quite separate. So this was this one species, Salaphora pupula, as it was then, in one pond in Edinburgh, and there are these 9 variants in this case. There are now more of them, because others seem to come in from time to time with geese and ducks probably. And each of these different types turns out to be a separate entity. Most of them are sexual species, and if you try putting them together in the lab or just watching what they do brought in from nature, they will not interbreed with each other. There are 3 that we don't know enough about. One here seems to be always vegetative. It seems to have a life cycle where it doesn't get smaller, so it disobeys all the rules that I was telling you about at the beginning of this presentation. This one is still a mystery, and this one is not a sexual species. We'll come back to that later. So we have got 9 things there that for one reason or another seem not to be able to interbreed, and therefore seem to satisfy the criteria for species according to the biological species concept. And when you look carefully at the morphological variation and also at molecular data, you find that they are separate on the basis of those 2. Just to follow this through a little bit more, let's just take one of those 9 things and see what happens if we go outside the Blackford Pond in Edinburgh. Old Reekie, by the way, gets its name from the city where I am at the moment, from Edinburgh, which used to be a very smoky place. And old is the Scottish way of spelling old, and Reekie refers to the fact that it smelt a lot and it was very smoky. So this is the old smoky place. That's why it's called Old Reekie. Okay, let's look at Old Reekies from elsewhere. It's no good just having a species concept that applies to one pond in Edinburgh. So what happens if we sample Old Reekie-like diatoms, so we take things that are morphologically similar, from a wide geographical range? Now, what we've seen previously is that what seemed fairly simple from one place got less simple and less satisfactory when samples were taken from elsewhere. But this doesn't happen with Salaphora Old Reekie. If we go around sampling similar ponds in different places, this is Edinburgh, this is Australia, we find that we can actually make a pretty good job of interpreting the variation in this species complex. First of all, we should, and we do, look at the morphology to see if we can come up with some boundaries between different types just based on what they look like. And on that basis, we were able, with all of these, for example, to say that these seem to form a continuum. They are the kinds of variation that you get during the life cycle, whereas this one is a bit coarser and bigger. And this one is slightly different again. So morphology begins to give us some hints that there are other things in the Slephora oldriki group. Then we can look at these organisms with mating experiments. These are a series of clones, single cells isolated from nature and grown up to form a culture, and then introduced to each other in pairs. So we take a cell, for example, from an Edinburgh pond. This one is from another pond in Edinburgh, and we can mate it with things from an Australian pond here, and you can see that the ones from Australia and the ones from the Edinburgh pond are able to mate. So we were able to show that there's a species, the real oldriki from Edinburgh, but it was also present in Australia, whereas there are others that apparently do not occur in the UK as far as we've been able to judge so far, and they form a separate group that are slightly different morphologically and separated in terms of their mating behavior. And when we look at molecular data, we find that that correlates also with differences, for example, in the RBCL gene. The southern oldriki, the one from Australia here, forms one group that is separate from true oldriki here and from various other very similar diatoms. And overall, up till now, we have come up with this whole series of what appear to be quite separate species, most of which don't yet have names because I haven't been able to complete the process. I think that every new species should be studied thoroughly, and so far I have not had time to do the scanning electron microscopy that I think would be necessary to complete the picture of these organisms. So a combined morphological, genetic, and mating approach point to boundaries between hierarchical and reticulate variation in this complex, and we can do that in whatever other species complex that we like. Of course, it takes a lot of time. In the last decade, furthermore, we've made the first progress in understanding. When I say we, I'm talking about the diatom community. I'm not talking about me necessarily. We've made a lot of progress in beginning to understand some of the cellular mechanisms that underlie the formation of these boundaries that stop gene exchange and so on. First of all, it was probably obvious to you, and you were probably thinking about it at the time, that for these sexual behavior to work, sexualized diatoms have to find each other. So these cells have found each other and mated with each other. These cells have found each other and are mating with each other. How did they do that? It wasn't by accident. Cells don't just kind of stick to each other at random. I can show that if you want. They have to find each other, which means there's signaling between cells. So here in a different diatom species altogether, this is a naidium, you can see that cells form big clumps. They have come together actively, finding each other through some kind of chemical recognition process. And there are now several cases where that process of signaling from one cell to another has been demonstrated in one form or another, and in one particular group the process is now beginning to be understood very well. This was one of the first ones, some studies that we made of what was then called Pseudostaurosyra traineri. It forms female gametes and male gametes, which I don't want to go into in detail about their morphology, but for that, for the reproduction of this organism, there have to be signals going backwards and forwards between the cells, and those signals ensure that the male gametes find female gametes. In the better studied system, which is one that involves a genus called Seminavis, they have been able to work out not only that there are pheromones, but they have worked out the chemical structure of them and how they begin to influence, how they influence the cell. So, for example, this Seminavis uses a chemical called L-diproline to signal from one cell to another to say, here, come and find me, I am ready for sex. So we could imagine, in terms of the biological species concept and the separation of species, that changes in signaling between cells, for example, changes in the chemoattractant, the pheromone that is involved in helping cells to find each other, or in the pheromones that sexualize cells, because there must be something that tells one cell that there's a cell nearby that is ready for sexual reproduction, that changes in these signaling processes may underlie speciation in some cases. The big missing link, and perhaps a challenge for somebody listening here today, is that, as far as I know, no one has yet tackled the extra problem of cell-cell recognition. Now, these cells have come together by some kind of active signaling using a pheromone to find each other. These cells have also found each other through some kind of signaling. But there must be something extra that tells these cells here that they have actually found each other, that once they are close by and touching, that one cell says to the other, this is the one to mate with. This is the same as me. You can see it perhaps in relation to this cell here, which belongs to a third species. This is Celaphora aldrichi, this is Celaphora capitata, and this is Celaphora blackfodensis. Capitata is mating with capitata, but it is touching blackfodensis. This is aldrichi is mating with aldrichi, but it is touching blackfodensis, and yet is not mating with it. So there's something that is telling these cells that they have found the right thing and they should mate with the cell they're touching, not the one over here, because they don't recognize that as the same species. So I think there is a missing link in our understanding at the moment that is a real challenge to somebody to find out something about this cell-cell recognition system, to know how do the cells know that the cell they're lying next to is the same or something different. Anyway, regardless of that, I think we could say overall that the evidence I've presented to you shows that splitting has a better foundation in diatoms than lumping. Better to split, it's more likely to be true than putting everything together once you have taken into account how diatoms vary during the life cycle and in response to environmental factors, because there is phenotypic plasticity in diatoms and we need to recognize that. Okay, so in terms of big hypotheses, this kind of feeds on to grander things in a way. Back in 1988, a very long time ago now, probably before some of you were born, there was a paper, an influential one by Robert May, which reckoned that there was a deficit of small species. So once you were smaller than a millimeter, there were fewer species there than there ought to be. So this is a log of cell size or organism size here. And for larger organisms, through from whales down to kind of millipedes and that kind of size of cell, there's a very good log linear relationship between the number of species and the size of the organisms in those species. But there's a deficit of species of smaller things. And the question since that time has been, is that real, or is it an artifact of our classificatory system? And you can see that as far as the diatoms are concerned, it does indicate, the evidence indicates there is a taxonomic problem, that we are not recognizing a lot of things that really ought to be called species. Okay, so if splitting has a better foundation in diatoms than lumping, what are we going to do about it? And are there any problems? There are some problems. First of all, there are various processes that are going to make life difficult. If we take higher plants as a guide, then there are problems, for example, of hybridization, which blurs the boundaries between species. There are automictic or apomictic groups, things that self-fertilize or don't fertilize at all, which multiply boundaries between species. You get lots and lots of species formed that are, by definition, unable to exchange genes. And there is always the problem of geographical separation. If we find that in the laboratory, two organisms can exchange genes, does that really matter if they are separated in nature by 2,000 or 20,000 miles? Okay, what about hybridization? Hybridization leads to blurred boundaries between groups of organisms. So we have here two Erica species. These are moderately distinct, except that from time to time they hybridize, producing something that looks a bit like both. Does that happen in diatoms? Yes, it does happen in diatoms. It's been known since 1958 that you can get hybridization between closely related, or rather morphologically similar diatoms. I've seen it myself in the kinds of species that I look at. So here we have two things that look fairly different in terms of their morphology, and yes, they can mate and produce a zygote. So this is a zygote here. This cell has fertilized that one. This has been investigated in some detail by Peter van Ormelingen in Eunotia, and he's been able to show that two parental types are able to hybridize and form a rather intermediate form. But what he found was that although these parental types can interbreed and form a hybrid, the hybrid was not very keen, as it were, on reproducing with its parents or itself. So there are kind of barriers to the gene flow between different lineages. The most recent and most comprehensive work in this area was published 3 years ago by Sam de Decker. He's part of the team in Ghent who do a lot of really superb work on the sexual reproduction and speciation of diatoms. And he was able to show that there are 3 clades living together. So there's the molecular data showing 3 separate groups here on the basis of the RBCL gene. These 3 occur together and they are able to interbreed to some extent. There is only partial separation between them in the laboratory. But what he wasn't able to conclude is what relevance that would have to nature. Do these organisms actually exchange genes to any significant extent in nature? We don't yet know the answer. So there are some reports of hybrids, but little information about their significance. Their existence has been demonstrated usually in the lab, but not normally in nature. Okay, what about organisms that don't have a normal sexual system? Now, again, if we take a lesson from higher plants or larger organisms, which are always better studied than diatoms, we know that problems can arise, taxonomic problems can arise in interpreting variation in groups that don't have a normal sexual life cycle. So, for example, in this type of compositae, Taraxacum species, Hieracium species, and others, they often have unusual mating systems, reproductive systems that involve the loss of or partial loss of sexuality and the formation of seeds without fertilization. And in those, that it is notorious that you find that almost every plant is different from the next. Well, not every plant, but every lineage, every small lineage is different from another. Those lineages may last for hundreds or thousands of years, but there are hundreds and hundreds of them, and they look very similar. And the problem becomes even worse if a group like this has some sexual forms, because those sexual forms recombine to produce new lineages, which then are perpetuated by apomixis or selfing. Does this happen in diatoms? Well, yes, it probably does. We already know that there are quite a lot of uniparental diatoms. In other words, diatoms that can reproduce and oxosporulate without going through a stage of pairing and fertilization. They just form oxospores on their own, sometimes with meiosis, sometimes without. These are examples of those, and some of these, such as the ones I'm showing here, occur in genera that are notorious for their taxonomic difficulty. Nitzschia is one of the most awful genera you could possibly hope to come across or dread to come across. The variation pattern is awful. Trying to understand it is difficult. There are almost no morphological characters to work with, and every time you isolate and grow something and sequence it, you find it slightly different. This is a case where, again, there are for sure a lot of apomictic or selfing organisms, and part of the taxonomic difficulty, I think, must be because of that. There are also apomictic forms in Eunotia as well, and this did some Peter van Ormelingen in a series of papers did some very nice work on these. So there is increasing evidence for uniparental auxosporulation. Meiosis may be present with the formation of two gametes within a cell, but those gametes fuse, so there is no fertilization from another partner. Meiosis may occur, but two haploid nuclei from the same cell may fuse. There may be avoidance of typical meiosis, or there may be no meiosis-like process at all. And all of these are likely to cause problems for interpreting what species are, because the homogenizing and recombining effects of sexual exchange are absent. So, summarizing all of this, I think we could conclude that we could answer, if we were Haldane, if we wanted to say something about the nature of God from a study of creation, we would answer perhaps an inordinate fondness. In other words, a huge liking for, a desire for more species of diatoms. Okay, finally, to kind of finish off before I let you say anything in case you want to, what do all of these diatoms do? Do they have different ecologies? What drives speciation? And do they have different geographical ranges? So I'll go through this pretty fast. First of all, if you can tell them apart morphologically, sometimes you can see that these very slightly different things, related very closely to each other, can have different ecologies. So here's an example where two morphologically different species in the same complex had a different seasonal behavior. One was a spring species, here the blue one, and one is a late summer autumn species. But this, of course, raises a practical question. How do we actually do any ecology on these things when they are so difficult to tell apart? I can't tell all of these apart, although I can make a bit of an estimate that this one, for example, is different from this one. This one has slightly wider ends than this one. But am I really going to be able to do that consistently? Is anyone else going to be able to use that criterion? This is where DNA metabar coding, and indeed DNA studies altogether, but particularly DNA metabar coding, come in useful. DNA metabar coding is a molecular technique that depends on using a short DNA sequence to tell species apart. And then we use that barcode, that short sequence of DNA, together with high throughput sequencing to obtain millions of reads of that barcode from environmental samples. So we take an environmental sample, we extract the DNA, we find out, we then pass it through a machine, an Illumina machine at the moment in our case, to produce millions of reads of this barcode so that we can see how many of each type of organism are present. And from that, using those kinds of data, we can begin to probe whether particular genetic variants have their own separate ecologies. So these are two diagrams. Those of you who are ecologically minded may understand these diagrams better. But basically, if you've got a color here, it shows some kind of association between the occurrence of a species and particular environmental factors. So here's Acnanthidium minutissimum group, not the species in the narrow sense, but the group of species allied to Acnanthidium minutissimum. This is a fistulifera. And you can see that the genetic variants, which are on the y-axis here, have different preferences. This group react in one way to these particular parameters, conductivity, calcium, and so on, whereas this group down here behave in an exactly opposite way. I've forgotten which way around it is. Oh yes, it said here. These react negatively, as it were, to calcium and conductivity, whereas these react positively to it. So we can see that different genetic variants have different tolerances or preferences to environmental variables. What about the biogeography of these splits, these very apparently minor variants we are actually thinking now are separate species? Well, again, one of the problems is how do we actually work that out? Here are 4 things from Australia, and here are 4 things from Scotland. Is this the same as that? Is that the same as that? And so on. Looking at the morphology, you might say, well, yes, I think those fit pretty well. We'll assume that they are the same. So we will say that all of these 4 can occur in Australia and Scotland. However, when you do molecular analyses, you find that 2 of them, yes, are the same. These are 2 Salaphora ulrichi. These are 2 what was then called Salaphora urban elliptical, which is now Salaphora bisexualis, whereas the 2 on the left are different. This one is not the same as that one. This is not the same as that one, despite the appearance that they are. Okay, so then if you have to use molecular data to prove that things are the same across geographical areas, then you've got problems because, of course, it takes an awful lot of work to isolate single cells and sequence molecular to get molecular information from them. And this is again where metabar coding in. We could take environmental DNA samples from ponds here and ponds everywhere, and without the need to culture diatoms and sequence them by the orthodox method, the Sanger sequencing method, we can work out whether diatom species are widespread or not. And I think this is what worries me a bit about a lot of what's being done at the moment. There is a huge amount of new species being described, and a lot of them are said to be endemics of various areas. And basically, how do we know that? If it is just done on the basis of a morphology and maybe a few sequences, how do we know that we have really surveyed the world to find out where they are and whether they are new or not? It really worries me. I think distinguishing and mapping species of microalgae, it's not a trivial task. Species are not trivial entities. They are not things just to be invented on the basis of a couple of weeks' work. Furthermore, one record in a particular place, one good record, establishes presence. But how do you establish absence? How do you know that something is an endemic in a particular area if you can't have confidence that it isn't actually somewhere else that you just haven't sampled? And this, I think, feeds on to some of the work that has now, in some people's minds, been abandoned. Nearly 20 years ago, the predominant model of biogeography for diatoms and other small things was this ubiquity hypothesis, which suggested that small things move around the world so quickly that you're not going to have endemics and you're not going to have very high species diversity. The ubiquity hypothesis is now generally rejected by many protist people, and particularly for diatoms. But that could be premature, because really we're not in a position to say what the distributions of diatom species are. And perhaps they are controlled more by ecology than by dispersal. You can get evidence that a particular diatom species does have dispersal constraints. We did some population genetics work a number of years ago that showed that dispersal is a problem for diatoms, that gene flow is not unrestricted across the whole world. It takes time for genes to spread from Scotland to England, from England to Belgium, from Belgium to Australia, and so on. However, if you look at a larger scale, you find that overall, there's not much problem for diatoms in spreading. So this is some wonderful work that was done fairly recently, well, 8 years ago now, on Pinularia borealis, which is a terrestrial species, and it showed quite nicely that there were a number of lineages that seemed to be restricted to particular places. So there was a Chilean lineage here, another one here, Belgian lineage, another Belgian lineage, and so on. So we have a number of lineages that seem to have some degree of specificity for an area. But then when you explore, think about it a little bit further, you can see that this clade here is in Mongolia and Chile. Well, they're quite far apart. This clade is in Antarctica, in the French Alps and Belgium. This clade is in Chile, Canada, and Czech Republic. So even if a particular clade is restricted to a particular region, when you go up in the phylogeny, or if you go up in the taxonomic hierarchy, you fairly quickly lose any biogeographical signal. That, to me, suggests that diatoms move around pretty fast. Okay, so we need, I think, DNA metabar coding. It gives high resolution, it gives consistent identification, it allows us to survey across a wide area accurately, and it's getting cheap. Last thing is, what drives speciation and maintains local species diversity? We have these genetic variants, and some of them are apparently environmentally related, but not all. We've already shown some cases where there seems to be no relationship between genetic variants and physicochemical variables. Perhaps, though, they relate to things like parasites. So it's been known for some time that some parasites discriminate very well between particular diatoms. And in Blackfoot Pond, for example, these different types of Salaphora pupula obviously can tolerate the same kinds of conditions, because they're all growing alongside each other at the same time, but they do react differently to parasites. And if you look at the percentage parasized by particular chytrid species, for example, different species are affected in different ways. And this applies to other species too. There are a huge variety of different fungal parasites, probably bacterial parasites too, and they affect things in different ways. So overall, I would say diatoms, there are an awful lot of species. We estimated back in 2013 that there might be 100,000. I think that is easily possible still, based on more recent data. So there's plenty of work to be done. I think it's exciting work, and I think there's so much more that we can do than just looking at the valves and the cell walls and classifying those. These are exciting organisms with real biology that's worth looking at. So thank you very much indeed. I apologize that I've left only a short official time for questions, but if you want to contact me at any time, I will try to answer, and I would be very glad to hear your feedback.
Thank you, David. Well, we have some comments here.
Okay, do you want me just to try to answer as I see them, shall I, or?
The second part.
Diatoms don't have identity issues, we create it. Yes, I guess that's true. The problem is that, of course, they are not interested in doing biomonitoring. We are. They're not interested in establishing biogeographical ranges. We are, perhaps, and they're not interested in phylogeny, and we are. So we have to find a way to find out from them what they regard as their own identity. That's my feeling.
You mentioned about hybridization in diatoms that is possible, whatever. And in this case, this species would not correspond to the biological concept of the species. The reproduction in the descendants of this hybridization is also possible.
As far as we know, certainly Sam de Decker's work, the recent work, has shown that some of the hybrids, certainly in the laboratory, seem to be not only viable, but they can back cross to the parent cells and cross between themselves also. The question is whether that is meaningful, whether it relates to what the diatoms do in nature. And to solve that, of course, one would be having to look at population genetic markers and explore just to what extent the natural populations are separated or not. But just to go back to what you were saying about the biological species concept, first of all, it would have to be shown that it has no relevance in nature. So that's what I kind of just covered. But also remember, if I could say so, that as I was indicating from the Dekerov article, there are, as it were, different levels of different processes going on during speciation. So, for example, two things might, according to that, the biological species concept, if you like, is only part of the story. And I do want to emphasize that. It's not that you could say, okay, I failed to find complete reproductive isolation, so these are not separate species. No, that doesn't work. That is one criteria. You found that the two things couldn't reproduce with each other sexually, no gene flow at all. I can't really see how you could say that those are still the same lineage. They have separated. They are now separate entities that will evolve independently. And that's the big problem about apomictic and selfing species, that there's no way for them to exchange genes, but the differences between them might be quite trivial. So you might not want to separate them taxonomically as separate. So within the case of things that can exchange genes, well, they may exchange genes, but there may be such selective pressure in nature that the hybrids that are formed have no permanent existence. You know, they are formed, well, maybe they transfer one or two genes from time to time in a, you know, but basically these two lineages have separated because there is an external pressure, the selection pressure, that is keeping those two lineages apart. So it's, I think, a question of looking at all the different things that happen during the process of speciation and trying to interpret what we see in nature on the basis of all of those at the same time, I would say.
Yeah. Well, sometimes it was common that people ask us, well, how much robust are some morphological character to separate some species? But today we can see that this is not one condition to separate species, actually, because as you show for us, some Celaphora are very, very similar, and we have not some morphological characters. Maybe some curves at the end of the valve, whatever. Then this is not more the point for taxonomical, for example, taxonomical paper research.
That's putting it, I wouldn't want to push people that far. First of all, for sure, you're going to get a more robust picture the more kind of careful morphometric analyses that you do. And I think when you can't do careful morphological analyses, you have to be particularly careful about the results that you publish. You know, that how much confidence you have in the decision. And if there's no absolute confidence, then it should be phrased that I think, you know, this is a taxonomic hypothesis and maybe it will be proved wrong. What was I going to say about it? My thought process has gone a bit. It will come back in a moment. This is one of the problems.
Yeah. We were talking about the robust characters.
Yes, right. The other thing was that every case is special. And that's, to some extent, also what I meant by the criticism about triviality. The idea that one can go to a particular species complex and sort it out by applying a set of morphological rules. Now, for example, if it's got more than 10 stripes in 10 micrometers, it's X. If it's got less than 10, it's Y, and those are separate. That may work in some cases, but you can't use that as a universal criterion. As an example, in most niches and their relatives, striation density is a pretty good character in my experience. Once you make a decision on the basis of overall the shape, the molecular data, mating data if you've got it, whatever you've got, you come to a decision. When you look at the striation density, you find that usually it varies very little within a particular species, as you've defined it. But in Samodiction, which is very closely related to Nitzschia, just a marine group with a different kind of valve structure, you find that the striation density there is much more variable. So whereas most niches perhaps vary by, you could say that something that's got 20 to 22 striations in 10 micrometers is likely to be different from one with 24 to 25. In Samodiction species that seem to be genuine species based on observations of clonal populations, the variation might be from 15 to 21 or 22 within what looks like a perfectly good species. So you just have to judge on the basis of the populations that you're studying. And I think that population approach is the one one really has to take. There is a tendency now, and I understand why, for people to go to type material and try to build up a picture of variation based on type material. In my opinion, and it's my opinion only, although other people might agree or disagree, but I think focusing on type material is a big mistake. The first thing to do is to look at natural populations that you have and you can sample from your area and as many other areas as possible. You build up a species concept from that using whatever evidence you have, molecular, mating, morphological, everything. And then you have, I hope, some ideas about what the species are. You've come to that decision about the boundary between hierarchical and reticulate variation. Okay, now you've got your entities. What are you going to call them? That's when you need the type material, and really only then. And the type material tells you how to apply the names that exist to those populations and species that you've distinguished. And if you find that what you've distinguished has no individual in it that corresponds to any type of any species described before, then you have a good reason for describing a new species. What people tend to do, I think, maybe it's unfair saying that this is what people do, is that they tend to think, if I've got a taxonomic problem, what I must do first is to go to the type material, because if I understand the type material, I will know how to identify the species. And up to a point, that's true, but it's impossible from the type material to get a clear picture of how that species varies. So I would always say, start, if you can, with much more material. Use the type material alongside that to interpret the outcomes.
Yeah, sometimes the people make mistakes, confound the concept, the typological concept of the species, and use the types as a concept of the
Exactly,
species.
exactly. And what you get as a outcome of that is that, in fact, according to the International Code, as you probably, I'm sure you know, the International Code allows people to designate populations on slides as types. So to validly publish a new species, all I have to do is to say that the type is on slide X, and slide X might have 1 or 10,000 individuals. They may be identical genotypically, so they've come from a clone, or they could be many different genotypes that we don't know about because it's come from a mixed natural population. And that can be a type. But it shouldn't be, because what we need to do for the type is to make sure that the type can be used to fix the name. And for that, it is only safe to specify one individual, or if it comes from a clone, okay, fair enough, you can specify the clone, the slide that has that clonal population on it as the type. But it's a big mistake to say the type is slide X, because slide X probably has a lot of variation that is genotypic and does not help to define the species name in a permanent sense.
Yeah. To complement, we have a question here.
How do I see the future of taxonomy based on morphological characters? Is there hope outside metabar coding? Well, of course there is hope, and we have to start with what we have. And morphology is the first stop, and it's I'm sure will for a long time be the major way that we study populations. But if we're going to define those species in a way that other people can use them, we've got to take some extra steps that most people are not taking at the moment. So you don't need metabar coding so much, for example, if you can really show people how convincingly how your species vary. People now try to do that in papers by having lots of photographs of the diatom, and that's excellent. It would be much more helpful probably if they contributed to a database that showed that variation online, and people could just pick up the photos from the web and compare them with what they have. That said, metabar coding, well, barcoding and metabar coding, metagenomics are the ways that people are going to go. And I think it's going to develop pretty fast. And the trouble is that we all get left behind as the technology changes. Those of us working on diatoms have been left behind in many ways by people working on higher plants and higher animals. They are already doing things that we will not be able to do for 20 years. They have been doing things that we are just starting to do now, and they have been doing them for 50 to 100 years. So the technologies we apply are always going to be changing, and we're always going to be playing catch up, as it's often called. The sooner that we can kind of try to work in the new environment, the better. So if you're describing a new species and it is not economically impossible for you to do some sequences, that would be great, because it will fix the use of your name and help other people to understand what you've got, because they can apply the barcoding in their own area, check that theirs is the same as yours. They can do metabar coding and know that that they can label particular genetic variants as your species, which otherwise becomes tricky.
You also said that some hypothesis that diatoms move quickly around the world, and use the example of Pinularia. Do you know, well, how, because I cannot see so many papers, studies, research, how these diatoms can move around the world, because it's the dispersion, the dispersion methods. Well, all the people hypothesize that birds or boats, whatever. But well, I cannot see a lot of research about that.
It's true. It's true. There are certain papers on birds. Kathleen Atkinson in the 1980s looked at the possibilities of bird dispersal. Of course, when you've got organisms that move around and kind of carry diatoms with them in an obvious way, like turtles, you can see which species are on the turtle and show what kind of possibilities there are there. Boats can be sampled, but surprisingly to me, they haven't been sampled so much. The hulls of boats, you know, not just the ones that the tankers and so on, but what about long distance yachts, as it were, that don't take so much care with perhaps taking the fouling organisms off the bottom? There's the question of ballast water transporting. We know they transport dormant cells of dinoflagellates around. Presumably they do that with diatoms too. So lots of possibilities, but little hard evidence. To me, the kind of key issue, if you like, is that it happens. How it happens, it would be very nice to know better how it happens. I was reading a book that possibly other people have read. Unfortunately, I've forgotten the author, but it was about biogeography of larger organisms. And what struck me was about the old world, new world monkeys. They have crossed the Atlantic. Well, if it's possible for a monkey to cross the Atlantic well after plate tectonics pushed the continents apart to the extent that they are not going to have swum, well, if those can cross the Atlantic, then is there likely to be a big problem for diatoms? And the genetic evidence, I think, for diatoms indicates that they really move around quite fast. There are some genera that seem to be restricted, like Eunophora, like what's the one in South America, in southern South America? I've forgotten its name. It'll come back sometime. There are some genera that seem to be genuinely restricted to particular continents, and the evidence makes it difficult to believe that they are isolated by ecology. So dispersals for those seems to be a problem. But for an awful lot of diatoms, I think, you know, the genera are all over the place, and they have been diversifying and giving rise to species that are also all over the place. So it looks as though they get blown around or they get moved by bird vectors or whatever fairly effectively. I think the first hypothesis has to be that they are separated by ecology, and it's only if you can show that things are really absent, that's always a problem, and are unlikely to be separated by ecology, that you can begin to think this is a dispersal-related endemic.
Great. We have another two questions. And a lot of comments here. Thanks, and that after I will put here to read. Okay. But first, the two last questions. Wait just a minute here.
Okay, are there works using machine learning for morphological identification of species? Yes, there are. And some of them are pretty impressive. We tried doing this, and we weren't the first. The first people who tried to do morphology, well, kind of automated identification using morphology, were a group in the United States who were using, what's it called, optical diffraction as a method of characterizing and identifying valves. Our approach was to do it on the basis of outline. We tried two variants of that. Pattern inside the outline was a bit more difficult. We did have a little bit of progress with that, but then everything stopped. There never seems to be any funding to implement anything. You get money for exploring the possibility of identification, but not how to actually develop a system that works. The best systems that I know are the ones that have been developed by the group formerly based in Bremerhaven, now in another site in Germany whose name I forget. The name is Bank Besterei, and Michael Kloster, and they have developed a really impressive system that works first of all on image. So they have now got it to the stage where they produce wholly in focus images, and then use those wholly in focus images as the basis for extracting data on outline in particular, to characterize individuals and therefore populations. I think there's great hope for it, but you need a lot of money. The reason why I can't implement that is that you need a motorized stage that on your microscope that works not only on the Z-axis, the depth axis, but also works on the XY, so it controls the whole of the stage. We've got a fantastic microscope in Edinburgh and another one in Catalonia where I work the rest of the time, but we've only got Z-axis control. So we can take lovely through focus images, but we can't motorize the stage to go and look for other diatoms, whereas the people working formerly in Bremerhaven and the other lab that now escapes me, they can do everything, and they produce these vast virtual slides that they can scan overnight, if you like, to get the picture of everything that's on the slide and find all the diatoms on it. And then the software will go and look for diatoms, identify them, characterize them, and so on. So overall, the thing is, there's great hope for automated identification, but you've got to have a lot of money. And you have to ask yourself also what the trade-off is between the money that you might spend on that and the money that you might spend on metabar coding, for example, depending on the purpose that you want to apply it to. Automated identification is not a cheap way to do a better way of surveying slides.
Okay.
What about the future studies of diatoms? Where can I start? I wish I had another 45 to 50 years. I get quite emotional, you know. There is so much to do, and what I think is encouraging and inspiring is that it really doesn't matter too much what level of resources you have, because you can design a first-class project with likely to get first-class results with really quite limited resources. It does depend, of course, on what your institute values. So my institute in Edinburgh never particularly valued diatoms, full stop. So when I retired during the pandemic last year, or just before it got bad, I was not replaced because the institute doesn't like diatoms particularly. And the only funding that I could have got successfully in the UK would have involved changing my direction completely to something very genomic. And I didn't want to do that. But there are so many things that you can do without going to genomics. If you have got culture facilities, you can isolate and grow things. And there's all kinds of things that one can do about mating and so on without doing molecular studies. If you've got that bit extra, you've got a molecular lab access to it, huge numbers of things that you can do with one gene still. Going back to the cultures, well, what else could you do with them? You could look to see whether the pattern of variation in their ecophysiological preferences in the wild corresponds in any way to what you have can detect in the lab. That's old-fashioned kind of work, but there still is a lot to do. Diatoms are just fantastic organisms. The sexual phase, I've spent a lot of time looking at the sexual phase and just how do they go through it. There's all kinds of chromosome work that has never been done. Never. There's only 2 people really in history who've kind of contributed in a major way to old-fashioned cytology of diatoms, using old-fashioned things like acetocarmine and Fulgenstein. Those are Austrians, Lothar Geitler, through from the 1920s to the 1980s. He was still publishing papers. And Elizabeth Chermak-Wurst, who was one of Geitler's students, I think, who did some interesting work on the nuclei of diatoms, not the chromosomes so much as the kind of nuclear structure, which again has never been followed up on. Or hardly ever. I've done a little bit, and Alka Purečková in Czech Republic has done some as well. So there's that can be done, you know. And what about devising a method to do diatom chromosomes? They have huge problems. They've got this beastly silica shell around the outside that stops you squashing the cells in the way that higher plant people do to get chromosome preparations. I'm sure there are ways around that. If you've got a friendly chemical laboratory where you could do hydrofluoric or ammonium fluoride digestions, both extremely unpleasant chemicals, but they take the silica out and maybe they leave the chromosomes intact. So you could take the shell off and start squashing cells and try to get chromosome preparations. And that opens the way to working out the chromosome evolution of diatoms. Perhaps it's not worth doing. Maybe we've moved past that. Maybe it's easier to work out chromosome numbers from genomic data, but it's possible and could be done. And somebody with some imagination and determination could do it. There are just so many things that could be done in diatoms. It's following up observations, having imagination, thinking about what you see, and not following what everybody else does necessarily. If I hadn't followed up what I saw when I was essentially doing systematics. So I told you, if you remembered, I looked at diatoms for the purpose of sorting out diatom genera in naviculoid diatoms to try to understand them well enough to produce accounts for what was basically an atlas of diatoms genera. In the process of doing that, I saw these mating diatoms and I thought, ooh, that might be interesting. So I followed that up, and that was the basis for the mating work that we did. And I think that influenced people like the group in Ghent, who have followed up and done much, much more impressive work than we did. So things grew from an accident because perhaps I took advantage of that accident and saw some potential in it. I hope it was like that. It was certainly exciting and I enjoyed it.
Yeah, it's very sad to know this history that you tell us, and at the same time is hopeful because it's our reality. Well, I don't know in the other countries from South America, but here in Brazil, it's our reality. We don't have a lot of professionals in the institutions working with diatoms because the public institutions don't have vacations about that, and we have problems with money and with tools, with to make some collect samples, whatever. But the major of these studies here are made by people that love so much this group, the phycology, the alga, and whatever. And it's grateful to know about that, because you are an example, people that have so many honors, awards, and work a lot, producing a lot of papers, a lot of research. And for me particularly, I'm an admirator for you so much. Yeah.
Well, that's very kind. Well, I don't know whether I hope it's encouraging also to people. I think it's great to work together, and most of my papers in the last 10 years, I think, have been very collaborative, working with 2, 3, 10 people. But I started without that. And nowadays, especially if you look at a lot of American groups, they work in a lab. There is let's say the Alverson lab, the Therio lab, and it's great. And these people do fantastic work, and they probably couldn't do a lot of that work without an intense work by a small group of people in a particular place. But it's not the only way that one can work. And I've never had a lab. I have worked physically with 1 or 2 people at the most in the lab at the same time. And nevertheless, I think it has been possible to make some progress. And it's always nice then to work with other people outside that 1 or 2. I enjoy that enormously, but it doesn't have to be a big lab. It is possible to keep on going independently, as long as you get a bit of encouragement from time to time from colleagues abroad or nearby.
Okay, well, now we have some comments here that I will put in the screen, and after we'll make the acknowledgements, okay? Carlos Wetzel sent some rug for you.
He's a great colleague.
Yeah.
Ah, Zilda, yes, hi.
Oh.
Well, thank you. Well, honestly, these are very kind. Oh, Luciel too. Okay, we'll have to pick up on the sex ratio sometime.
Well, as I said, it was an honor to receive you here with us and to learn a lot with you, with your lecture. And I would like to thank you again, and to send a lot of hugs there in Edinburgh, and also wish you health, because you have contact with someone with COVID. Okay, can you tell the last words?
Yeah, it wasn't just COVID. I've just 2 weeks before I tried to travel to the UK, I caught, well, not caught because it's already with me, herpes zoster. You perhaps know. So
Yeah,
I still have the relics of that, head and right eye. But honestly, it was a privilege to do this. It did take me some time. I always leave things too late, so I was still working to get everything ready this morning. But I'm so glad that you gave me the opportunity, and best of wishes to everybody. By the way, I don't want to imply that diatoms are the only things that are worth doing in the world. I wouldn't claim that I'm an expert on anything else at all, and I wouldn't claim that I'm an expert on everything in diatoms. You get left behind as time goes on, it's impossible to keep up with all the literature. But I find all the other groups of algae interesting, except I have to say, I don't know the multicellular groups well enough. They're just too complicated. But anything with one cell is fantastic. Euglenoids, dinoflagellates, even unicellular green algae are not too bad. And all of these fantastic new groups of straminopiles, I've discovered some of them for myself only through Metabar Coding data. We did a Metabar Coding survey of a marine sample or two. And lo and behold, though the reeds were supposed to be all diatoms, there were some fascinating other things as well I had not actually come across previously. I had to look up the literature and find that Bob Anderson had been doing some more really good work, and that was a treat. So every time you do something, there's something new and there's something to learn. So, well, thank you very much for this chance.
Okay. And in the afternoon, we will have with Mariangela Menezes a class about cryptophytions. And,
they're good too.
Yeah, and euglenoids also. Well, pessoal, até mais então. Fiquem bem. E eu espero vocês à tarde, tá bom?
Yeah. Adeu. Catalan.